This video will cover enzymes, including their nomenclature and classification, specificity of action, isozymes, mechanisms, kinetics, and regulation. Key concepts like the Michaelis-Menten equation, Km, Vmax, and Lineweaver-Burke plots will also be discussed. An enzyme is a protein or RNA produced by living cells, functioning as a highly specific and catalytic macromolecular biological catalyst. Enzymes enable efficient and specific chemical reactions in organisms under mild conditions. Enzymes are classified into 6 main groups based on the catalyzed reaction, as determined by the Enzyme Commission in 1961. Nomenclature is derived from substrates or catalyzed reactions, typically ending in AC and assigned an EC number with 4 elements. Enzymes are further classified into subgroups and subclasses based on functional groups or bonds in substrates. They are also categorized by molecular composition into pure enzymes, consisting only of protein, and binding enzymes, which require protein and cofactors for catalytic activity. Enzymes are classified into 7 categories based on the type of reaction they catalyze: oxidoreductases, transferases, hydrolases, lyases, isomerases, ligases, and translocases. Oxidoreductases, transferases, and hydrolases are the most abundant. The NCIUBMB enzyme list provides a functional classification system based on substrates and products of the catalyzed reaction. More detailed rules and guidelines for enzyme nomenclature and classification are available online. Enzymes are identified by EC, Enzyme Commission numbers, divided into 7 major classes based on the reaction type. The 7th class, translocases, was added in 2018. The table illustrates the 7 enzyme classes and the reactions they catalyze. The EC number consists of 4 components separated by full stops: class, subclass, sub-subclass, and a serial number. These numbers provide specific details about the reaction and the groups or compounds involved, facilitating enzyme identification and classification. This image shows the 7 EC classes: oxidoreductases, transferases, hydrolases, lyases, isomerases, ligases, and translocases, along with some subclasses for ligases. Now, let us discuss the specificity of enzyme action. Specificity is an enzyme's ability to select a precise substrate from similar molecules. This molecular recognition mechanism relies on structural and conformational complementarity between the enzyme and its substrate. Enzyme specificity is crucial for diagnostic and research tools. Some enzymes show absolute specificity, catalyzing only one reaction, while others are specific to a particular chemical bond or functional group. There are generally 4 distinct types of specificity. The 4 main types are absolute, group, linkage, and stereochemical specificity. Absolute specificity means catalyzing only one reaction. Group specificity targets specific functional groups. Linkage specificity acts on a type of chemical bond. Stereochemical specificity targets a specific isomer. Enzymes show different degrees of specificity, including relative or bond specificity, where the enzyme acts on substrates similar in structure and containing the same types of bonds, like amylase or lipase. Moderate structural or group specificity means the enzyme is specific to the bond type and the surrounding structure. Examples of moderate specificity include various peptidases like pepsin, trypsin, and chymotrypsin, which hydrolyze peptide bonds based on the amino acids involved, and aminopeptidase and carboxypeptidase, which act on peripheral peptide bonds. Absolute, high, or substrate specificity means the enzyme acts only on one substrate. Examples include uricase, arginase, carbonic anhydrase, lactase, sucrase, and maltase, each acting on a single specific substrate. Optical or stereospecificity means the enzyme is specific not only to the substrate but also to its optical configuration. Examples include L-amino acid oxidase, D-amino acid oxidase, and different glucosidases acting on specific glycosidic bonds. Dual specificity involves 2 types: an enzyme acting on 2 substrates by one reaction type, like xanthine oxidase, or an enzyme acting on one substrate by 2 different reaction types, like isocitrate dehydrogenase. Isozymes, or isoenzymes, are enzymes that differ in amino acid sequence but catalyze the same reaction, often showing different kinetic or regulatory properties. They fine-tune metabolism to meet tissue or developmental needs. Allozymes are variants from different alleles of the same gene, while isozymes are from different genes, but the terms are often used interchangeably. Isozymes were first defined by Hunter and Markert in 1957 as different variants of the same enzyme with identical functions in the same individual. They can result from gene duplication, polyploidization, or nucleic acid hybridization. Over time, these variants may become specialized to different functions, such as being expressed in different tissues. Allozymes, resulting from mutations, face three possible outcomes. Most likely, they are non-functional and removed by natural selection. They may be selectively neutral if the mutation is in an unimportant part of the enzyme. Rarely, the mutation may increase efficiency or catalyze a different reaction, leading to an increase in fitness. Next, we will explore the mechanisms of enzyme action. Enzymes catalyze reactions by binding the substrate to the active site. This binding changes electron distribution in the substrate's bonds, leading to product formation. The products are released, regenerating the enzyme. The active site's unique shape ensures specific reactions with one or a few similar compounds. The lock and key theory, proposed by Emil Fischer, explains enzyme specificity using an analogy where the enzyme is the lock and the substrate is the key. Only the correctly sized and shaped key fits the lock's active site to initiate the reaction. The induced fit theory, a modification of the lock and key model, suggests the enzyme is partially flexible and the substrate influences its final shape. This explains why only the proper substrate can induce the correct alignment of the active site for the reaction to occur. This image visually represents the concept of enzyme-substrate interaction. Enzymes exhibit varying degrees of specificity. Understanding the active site, particularly the amino acid side chains, is crucial. The active site of carboxypeptidase A illustrates the principles of substrate interaction. This figure visually illustrates the structure of a carboxypeptidase A active site. The substrate interacts with the active site through various bonds, including opposite charges, hydrogen bonding, hydrophobic interaction, and coordinate covalent bonding to a metal ion activator, such as Zn. This interaction activates the carbonyl bond, leading to the reaction. Enzyme kinetics is the study of enzyme-catalyzed chemical reactions. It involves measuring the reaction rate and investigating the effects of varying reaction conditions. Kinetics studies reaction rates, which for enzymes is the product formation rate. Enzyme kinetics helps reveal the catalytic mechanism, metabolic role, control mechanisms, and the effects of drugs or agonists. Enzymes accelerate reactions by lowering the activation energy, having a high affinity for the transition state. This means less energy is required, allowing the reaction to occur more easily and quickly. The enzyme facilitates the formation of this transition state. The transition state is unstable and transiently converts to a more stable product, which the enzyme then releases. The slowest step, or rate-limiting step, in enzymatic reactions is typically the conversion of the enzyme-substrate complex to the product, setting the pace for the entire reaction. The concentration of the enzyme-substrate complex changes until equilibrium or a steady state is reached, where the concentration remains constant. Enzyme reactions have three kinetic stages, with the short pre-steady state phase quickly transitioning to the steady state, which is used for measuring initial rate in Michaelis-Menten kinetics. Michaelis-Menten kinetics uses two important terms: Vmax, the maximum reaction rate at substrate saturation, and Km, the substrate concentration at half Vmax, which measures enzyme-substrate affinity. The Michaelis-Menten equation is used for steady-state kinetics with one substrate. The Michaelis-Menten equation describes how the initial reaction rate is affected by initial substrate concentration. It only considers the start of the reaction, ignoring the reverse reaction. While the rate versus concentration plot is a hyperbola, the Lineweaver-Burk plot offers a more useful linear representation. The Lineweaver-Burk plot allows for easier interpretation of kinetic quantities, such as the effect of an inhibitor, which can shift the line upwards by decreasing the Km value. Finally, we will look at the factors that affect the rate of enzyme-catalyzed reactions. This slide is a visual break before continuing the topic. The rate of enzyme-catalyzed reactions is influenced by several factors: enzyme concentration, substrate concentration, product concentration, temperature, pH, and the presence of inhibitors, activators, and cofactors. Generally, the initial reaction velocity is directly proportional to the enzyme concentration, assuming the substrate concentration is much higher than the enzyme concentration. The effect of substrate concentration is described by the Michaelis-Menten curve. High product concentration can inhibit the reaction rate, as the enzyme must release the product before binding a new substrate. Product inhibition is one of the most common ways to regulate metabolism. Enzymes have an optimal temperature where the reaction rate is highest, usually around 37 degrees Celsius in humans. Temperatures too low will lower the rate, while temperatures too high will cause denaturation and a loss of activity. Every enzyme has an optimal pH where it shows maximum activity. Most intracellular enzymes have an optimal pH near neutrality, 7.4. However, some enzymes, like pepsin, have highly acidic optima.